Every day, the kidney filters nearly 200 litres of fluid from our bloodstream. Functions of the kidneys include regulating the total water volume in the body and the concentration of solutes in that water, excreting metabolic wastes and foreign substances such as drugs and toxins, and producing erythropoietin and renin. The blood supply enters and exits the hilum of the kidney, along with the ureter, which carries urine to the bladder, where it is stored until it exits the body via the urethra. In a frontal view, the kidney consists of an outer cortex and an inner medulla region. Within each kidney, approximately 1 million nephrons are situated. The nephron is the functional unit of the kidney, so let's take a closer look. The nephron consists of two parts, the renal corpuscle and a renal tubule. The renal tubule consists of the proximal convoluted tubule, the loop of Henle, also called the nephron loop, the distal convoluted tubule, and the collecting duct. The collecting duct serves multiple nephrons. The renal corpuscle consists of the glomerulus and the Bowman's capsule, otherwise known as the glomerular capsule. Note that the nephron is surrounded by a capillary network. In 85% of nephrons, called cortical nephrons, this capillary network makes up the peritubular capillaries. However, there is a smaller number of juxtamedullary nephrons, which have long nephron loops, and in these nephrons, these capillaries are called the vasa recta, recta meaning straight. Blood moves from the renal artery through the medulla and up into the cortex, reaching the cortical radiate artery, before moving to the afferent arteriole and through a fenestrated capillary system called the glomerulus. Draining the glomerulus is the efferent arteriole, which has a smaller diameter than the afferent arteriole, allowing the glomerulus to maintain across its length a hydrostatic pressure of around 55 millimeters of mercury. Surrounding the glomerulus is the glomerular capsule, otherwise known as the Bowman's capsule. This capsule consists of a parietal layer, which is a single layer of epithelial cells, and a visceral layer, which coheres to the glomerulus. This visceral layer consists of branching epithelial cells called podocytes, which have little processes. The slits between the processes of the podocytes are called filtration slits, and they contain a thin membrane across them called the slit diaphragm. The hydrostatic pressure inside the glomerulus forces fluids and solutes through the fenestrations of the capillaries, through a basement membrane and the filtration slits of the podocytes into the capsular space. This process is called glomerular filtration. About 1/5 of the plasma that moves through the glomeruli each minute is forced into the glomerular capsule as filtrate. Cells and plasma proteins are too large to move through the fenestrations. This blockage is reinforced by the negatively charged basement membrane. It is the osmotic pressure provided from these proteins that allows reabsorption of water from the nephron to the surrounding peritubular capillaries. Glomerular filtration creates a cell and protein-free filtrate, which moves into the tubules of the nephron. It is within these tubules that essential chemicals are recovered in a process called reabsorption. Chemicals such as drug metabolites, which are attached to carrier proteins and therefore not part of glomerular filtration, can also be secreted into the tubules from the peritubular capillaries. The majority of reabsorption occurs in the proximal convoluted tubule. Glucose is reabsorbed through the apical membrane, co-transported with sodium. Glucose then moves through the basal membrane through its own transporter, while sodium is transported via the sodium-potassium pump. Potassium returns to the interstitial fluid via a potassium uniporter. It is the active removal of sodium from the cell that creates the electrochemical gradient that favors sodium entry at the apical side. Water moves due to osmosis through dedicated channels called aquaporins, while urea takes advantage of the paracellular route. The removal of water increases the concentration of the solutes in the filtrate and encourages their movement out of the proximal convoluted tubule. Chloride, potassium, magnesium, and calcium exit the proximal convoluted tubule via paracellular diffusion. Bicarbonate ions in the proximal convoluted tubule bind with hydrogen ions and are converted to carbonic acid, which is converted to carbon dioxide and water by the enzyme carbonic anhydrase. Carbon dioxide diffuses across the apical membrane where it meets with water, and the process is reversed again, facilitated by carbonic anhydrase. The hydrogen ions are returned to the tubule via sodium-hydrogen ion antiporter. The sodium that enters the epithelial cell then co-transports into the interstitial fluid with the bicarbonate ions via facilitated diffusion through the basal membrane. Although it doesn't anatomically follow the proximal convoluted tubule, we're going to skip ahead to the ascending loop of Henle. Here, secondary active transport occurs of chloride, sodium, and potassium via the sodium-potassium 2-chloride co-transporter. A sodium-potassium pump operates at the basolateral membrane, which creates the ionic gradient driving the co-transporter. While over on the descending loop, the presence of those solutes exiting the ascending side creates an osmotic pull, driving water out of the nephron through aquaporins, in the process increasing the solute concentration in the nephron, creating the gradient which drives solutes out of the ascending portion. Moving next to the distal convoluted tubule. The kidney has mechanisms for fine-tuning the body's water balance. One of the indirect mechanisms uses the steroid hormone aldosterone, which is secreted by the adrenal cortex in the adrenal glands. Aldosterone regulates the reabsorption of sodium by causing the synthesis and retention of apical sodium and potassium channels, and the basolateral sodium-potassium pump in the distal convoluted tubules and the collecting ducts. Water follows sodium via aquaporins due to the osmotic pull. Blood volume and hence blood pressure is therefore regulated via this system. Potassium is secreted into the lumen of the nephron. Depending on the hydration state of the body, the nephron can produce a large volume of dilute urine or a small volume of concentrated urine. This process occurs due to two factors. The first is that the interstitial fluid of the renal medulla increases in osmolarity from isotonic near the cortex to hypertonic near the apex of the renal pyramid. The second is anti-diuretic hormone, which causes an increase in the synthesis and insertion of aquaporins into the principal cell's apical membranes. An increase in aquaporins increases the amount of water removed from the collecting duct, pulled by the hypertonic interstitial fluid. Anti-diuretic hormone also increases urea recycling, which strengthens the medullary osmotic gradient, causing the further concentration of urine.